Okay, our class started, and here is that couple of seminars tomorrow and next Friday, and of course, these are optional, you don't need to attend them, but some students that are interested in biotechnology would be very interested in them. And in general, it's interesting because these are scientists that are actually professors at Northeastern that work at USM now because they're part of this new institute that's come to Portland called the RU Institute, and we're doing collaborative interactions with them, and it's really pretty fascinating. So it's going to be in this room tomorrow and then next Friday. So just interesting stuff if you're just hanging out with nothing to do. And some of you have nothing to do because you finished your term papers. Yay. I've heard from several students, term paper done. So what are we working on? Working on your term paper. When you finish it, send me an email so I know you're done. And I will eventually respond to you and say, yep, I checked it, got it. And then I also will read it, and when I'm done reading yours, I will send you comments on the grade. It's going to be, it was really foolish for me to make such an assignment because I just have so many things that are kind of at the emergency level that it's just silly. So then this is just a reminder to make sure you go over answer key video 2, and then also a general reminder of everything's got to be reviewed for our final. So everything we learned about, we covered on our final, and we have our final a week from Tuesday in this classroom. Yep. And so that's where we're at for our course. And we're going to keep rolling forward. You don't need to look at our book anymore for the material. Some of it's covered somewhat in your book, and we're just going to keep adding some stuff, and that will be included on the final. So Sam talked to us about that as CAR T-cell therapy for the 4th time, and it was more about the patient and less about molecular biology and cell biology. But you see a great topic to focus on. And a great way to learn stuff to remember, oh yeah, I remember in college this one guy in our class stood up and talked about that one cancer therapy and how they manipulated the gene, and then they made a receptor, and they put that transgenic receptor into the patient's own immune cells, and they put the immune cells back in so it would be more easily activated and attack cancer cells of a specific type of cancer. So great, and thank you, Sam, for that. And so then that's the last reading we've done, and we're good with that. And so we're going to roll on. Do people have questions? Because we are also reviewing for the final exam. So if people have any questions at all about anything at all. And you can interrupt any time, and it can be from anywhere in our course. You could be like, oh, I was looking back at the first exam and I have a question, about it or anything else. Yep, anytime. Just if you have a question, it's distracting you, so just raise your hand and ask it, and we'll get it taken care of. And if it seems like it's out of place, don't worry about it. It's my job to fit into our course. So I just want to describe just a couple of terms as we move forward. So these are just a couple of very simple terms about the structure of DNA. You may be already familiar with them, but just so we have a clear understanding of something, okay? And so here's that famous double-stranded DNA molecule, and it is a double-stranded helical molecule, and we can untwist it like that, right? And when we untwist it, it looks a lot like a ladder, where we have the nucleotide base pairs as the rung of the ladder, and then each of the strands is held together by covalent bonds that look like sort of the legs of a ladder. And if you look at the DNA molecule, it can be split into 2 molecules. It's really 2 molecules that are held together in these base pairs by non-covalent interaction. So we can unzip it or zip it back together. And if you look at the 2 parts, the 2 molecules, they're really exactly the same chemistry, but they're said to be anti-parallel. And so that's designated here, where it's using chemistry terminology to designate the orientation of the strands. And you see that 5' and 3', and that's just designating organization to it. I always tell classes it's kind of like these are built by Lego blocks, and a Lego block is a base and it has knobs on the top. And if you look, this strand is put together by Lego blocks in one orientation, and that's the opposite orientation. And the chemical nomenclature is just called 5' or 3'. And you'll understand that nomenclature really well if we just look at one of the strands, because here's one of the strands, it's called the backbone, and it has the carbons in the ribose sugar number. And so with carbons in organic molecules, you number the carbons, and they're prime numbers because the nucleotide base is got regular numbers, 1, 2, 3, 4, 5, 6, 7, and so those are numbered over there, so these have to be prime. As you can see, the 5' comes from that orientation, where there's a 5' carbon here, and that gives us an orientation, and there's the 3', and you can see the orientation goes 5' to 3', like that. So there's just that terminology we just want to be familiar with, and it's just a way of labeling. So because of that, scientists have a convention, and anytime they write down a sequence of DNA or RNA letters, the convention is they won't write down both strands, they'll just write down one, because the other strand is just the complementary base pairing, and they'll always write it the 5' to 3' orientation, always. And so most of the time they don't even label that, but it's always 5' to 3' that we're looking at, and it's because of that orientation. The other thing that's just an interesting detail is that all the DNA on our planet gets synthesized by an enzyme called DNA polymerase, and all the RNA gets synthesized by enzymes called RNA polymerase, and all of those nucleotide polymerases on our planet only can synthesize in the 5' to 3' orientation. So this strand got synthesized in that direction, and that strand got synthesized in that direction. So in addition to writing out 5' to 3', we're also writing the orientation that a polymerase would be making that strand. Yep. So just what we're putting there is, you don't need to remember the structure or anything. You just recognize right away, oh, I see that numbering system, because I'm sure you learned about that in chemistry. The numbers, and then, if you have another ring structure, you're going to have to label those with prime numbers. So that's where the 5', 3' comes in. And again, it's just orienting them like they're Lego blocks, and you're going to stack them together, and you can only stack them in one orientation. So now we go on, and this is something I just added some terms to because we'll spend the last part of our semester focused on the reality of gene expression being an extended pathway. So we talked about that some. And I just wrote the terms over on the side. So this is a figure that I showed before that's just from the intro biology textbook. And it starts out with the chromosome, the DNA, inside the nucleus in a eukaryotic cell. And we're inside the nucleus, and the first step is transcription. So we spent a lot of time talking about transcription regulation and the transcription factors, the proteins that regulate that transcription. Yep. So there's a lot of regulation. There could be regulation at later points in the pathway to gene expression. So the next is inside the nucleus, there is this post-transcriptional regulation, and people rarely say post-transcriptional regulation, they call it RNA processing. And so we said we're going to start out with a primary transcript that's made from the DNA by RNA polymerase, and then what leaves the nucleus is called the mature transcript, and in between we have these steps of RNA processing. And those can be regulated. You say most of the time it's almost like just an automatic thing. They get processed and they leave the nucleus. But there are potential regulation there, so we want to kind of pause there and learn about that some today. Then we saw exiting the nucleus. That's actually a regulated step. And now they're in the cytoplasm. And if this is a coding gene, it's going to code for a protein. So the messenger RNA in the cytoplasm will bind to the ribosome. It'll get translated. And there's translation regulation, so sometimes the mRNA doesn't get translated very well. And then there's this post-translational regulation. And we've actually talked a lot about post-translational regulation because it's any time you're regulating a protein, that's post-translational regulation. And so we have a really good example of post-translational regulation, that's phosphorylation. It turns off or on a protein, and thereby it turns on or off the gene activity. Yep. Then we learned last time about ubiquitination that targets or recruits a protein for entry into the proteasome trash can that gets it degraded to amino acids. So those are a couple of good examples. And we learned about them earlier in our course because we learned, P53 is unstable, beta-catenin is unstable, MYC is unstable, and they're all unstable and it's regulated like that. So that's how they're unstable, is they get ubiquitinated primarily and go into the trash can. Yep. So where you just want to spend some time on these two parts of gene expression, So today we're going to talk to them about this RNA processing that happens inside the nucleus. And then we'll talk a little bit about post-translational regulation. And we'll add a little bit of knowledge to that. But mostly we'll remind you, oh yeah, we've talked about examples of that. Yep. So questions about that?
We've talked a lot about post-translational regulation in the context of like phosphorylation, ubiquitination. Would we also categorize things like glycosylation and other like modifications of the polypeptide chain as that kind of, that happen in the ref ER?
Yeah.
That's all part of the same?
Yeah. Yeah, which is a good time for me to bring up this. So there's post-translational regulation, and as a subset of that, there's post-translational modification. And post-translational modification is when the protein has a covalent bond added or removed from it. So a modification, really we're changing the protein. And so phosphorylation is an example of post-translational regulation, and more specifically, post-translational modification. And so, Sam, you just asked about a couple other examples of post-translational modification. We can add things to the protein, covalently bonded to the protein, or remove them. We can also cut the protein. So, for example, when scramblase gets activated by caspase, it gets cut, right? It had this little inhibitory part of the protein on it, and that gets cut by the caspase and activates it. That's considered a post-translational regulation. We're going to turn it on, and specifically a post-translational modification. There are more examples of post-translational regulation than just modification. And there are many of those. Here's a great example we learned about a post-translational regulation, and that is estrogen receptor and estrogen hormone. That's not a modification, right? That estrogen hormone can bind or let go, but it is a really good example of post-translational regulation. Yep. And there's a lot of that. There's a lot of that. So we open up a big door, and it's a step in regulation of gene expression. We like that, right? Yep. Yeah, okay. Other questions people have about the layout? It's a nice one, right? Because it's very visual, right? We could say our classroom is the nucleus, and that's a nuclear pore, and we could have students get up and do all the transcription regulation here, right? Line up the desk, and we should do that today in a really noisy way. Line up all the desks, that's the DNA template, and so is going to be the RNA polymerase, and so is going to be transcription factor. And we get the RNA transcribed, and the students are holding hands because they're RNA molecules, and then they're going to go through processing over here, and then they're going to exit the nucleus, right? And there's a ribosome out in the class. I'll be the ribosome. I want to be a ribosome. I always want to be a ribosome. I had a dream about a ribosome when I was a child. And I just was like, I just feel like a ribosome, right? So we could set that up. It's very physical, and it's very conceptual in a physical way. So that's one reason to show this, right? We're in the nucleus, and we're transcribing the gene. We talked about our transcription factors. They're going to get synthesized in the cytoplasm. They got to get back into the nucleus to do their job. So it's very physical all the time. And I hope that helps with learning. So we're just going to be inside the nucleus, and we'll have transcribed the gene, and now we're talking about this RNA processing, okay? And so this is post-transcriptional regulation after transcription, and it's inside the nucleus, and it's called RNA processing, okay? So again, the terms, when we first transcribe the transcription unit, we get a primary transcript. That's also called pre-RNA by some people, but primary transcript is just a great term. And it's the same length as the transcription unit. So probably a good idea just to draw that out so everybody appreciates that physicality of it. So, so many times we've drawn out, here's a gene, and here's the transcription unit, there's the promoter, there's the enhancer, and the primary transcript is exactly the same length as the transcription unit. Of course, the primary transcript is RNA. And again, some people like to call that pre-RNA, pre because it's not yet processed. And then we know the transcription unit is made of DNA. So we get that made, and next RNA processing occurs, okay? All right, and so for coding genes, remember coding genes code for proteins, they've got coding regions, and there's a whole bunch of other genes that don't code for proteins, those are called non-coding genes. So for coding genes, these are the main types of RNA processing that happen in the nucleus. And again, much of the time, they're just automatic. They get done and we move on. But they're examples of regulating these. Yeah? So the first one is called the 5' cap. That's the only reason, today, that's the only reason why I mentioned the 5' term. You know what end we're talking about. It's right at the beginning. And a 5' cap is added. A lot of times the 5' cap is added as the gene is being transcribed. Okay? Then the second one is called a poly A tail, and that's going to get added after transcription. It's going to get added to the 3' end. And so I'll just foreshadow to our next slide so you can see that. So here's just an image of primary transcript that's getting processed. So the 5' cap gets added, and then the transcript gets cut, and this poly A tail gets added. So that's what we're talking about. See, 5', 3'. That's the only reason we talked about this structure, okay? So then we'll go on and we'll talk about this RNA splicing, and then an example of RNA interference that happens inside the nucleus, and we'll distinguish it from RNA interference that will occur after the RNA leaves the nucleus. Okay? So then we're going to roll through that, and that'll be our topics for RNA processing. So again, down here, these non-coding genes also have RNA processing, and we won't really dwell on those. We'll focus on these coding genes especially. Yep, okay? So the first one is this 5' cap, okay? So there's the chemical structure. Of course, you don't need to know it. It's actually a guanine nucleotide, guanosine nucleotide, and it's just stuck on, and it's really just automatically added. Okay, and so here's just a little statement about the 5' cap. There's a little zoom in on it. You can see it a little better. It's the regular nucleotide is just added in a backward direction. No big deal. Of course, yeah. So 5' cap, crucial modification of 5' end of messenger RNA molecule. This cap plays a role in protecting the RNA from degradation, facilitating its export from the nucleus, and promoting translation. So that's what it's there for. It's really, the 5' cap is really boring. It just gets added automatically, but that's what it does. And the most important thing it does is it protects the RNA from getting degraded. RNA is really unstable, and if it doesn't have a 5' cap, an exonucleus just chews up the RNA really fast. So it's really there just to protect the RNA, but you can see we'll mention it in the context of getting out of the nucleus, because you're like, you are an RNA and you're ready to leave, And one of the things you have is you have a 5' cap, okay, you're good to go. You have other features too. And then the final thing is it's also important, essential for the ribosome to recognize the RNA, and we'll have a picture of that. Yes?
I think so. Like the 5' cap is needed to leave, but it's not like immediately you're leaving. It's going to go through all
Right.
the next.
Yeah. You have to have your mittens on to get out of the nucleus, but you're also going to have to put your coat on and your boots on. Yep. And so we're inside the nucleus, and we're talking about just that 5' cap. It's really a boring one. Then the next one is this poly A tail. And poly A tail is just a bunch of A's that get added. They're not in the DNA. They get added to the RNA after transcription. So it is a part of the RNA processing, post-transcriptional regulation. And it helps stabilize the mRNA, facilitates export from the nucleus, and is important for translation. Same kind of role as the 5' cap. But it becomes more significant than the 5' cap. And here's how. The poly A tail is pretty long. It's usually 100 or 200 nucleotides long. And over time, it gets chewed up. And when it gets all chewed up, the RNA gets destroyed. So all of the RNAs are unstable, and the biggest reason they're unstable is the length of their poly A tail. So when we think poly A tail, we think, yeah, you gotta have it to get out of the nucleus, gotta have it to get translated. Also, especially important is, how long is this RNA gonna last? Okay?
The longer the tail, the longer.
Longer the tail, the longer it lasts, okay? And so let me just show you this, c-MYC, right? We know c-MYC is unstable RNA. So let's just look at this. c-MYC mRNA, known for its short half-life, has a poly A tail that plays a crucial role in its translation and stability. This tail is a long sequence of A's added 3' end. And it says down here, c-MYC mRNA exhibits a short poly A tail and is highly unstable. So, like, right, we like talking about this stuff that really was early in our book, because at the end of our course, we have examples, and we're like, yeah, you guys remember, we talked about MYC. The cell does not want to make MYC. One way it avoids it is the RNA is unstable. It just lasts for a few minutes. Yeah, you make it, yep, it gets out of the nucleus, but it's unstable. How is that unstable? It's an example of RNA that just gets a short poly A tail. And it's like, good luck, loser. You know, I'm done adding poly A to you. Yep.
So can the 5' cap not be degraded over time?
It does. So it can get removed and can be a trigger for degradation, but it's not anywhere near as important of a story in regulating gene expression as the poly A tail. And what's going on there to think about is, when we talked about transcription, we talked about how the cis-regulatory region, it controls where, when, and how much of an RNA gets made. Now we're talking about degradation. And so there would be a kind of a balance, right, between production and destruction. And so, how long your RNA is functional depends on both of those things, that balance, right? As for mRNA, we don't make very much of it, and we should chew it up really fast. And a big important way is we just don't add very many poly A tail to it. Yep.
Is the tail added after, or is it part of the transcription unit?
It's added after, so it's post-transcriptional regulation. Yep. And it's happening inside the nucleus. And here's a picture of what happens, okay? So this just shows, it's a nice picture. Oh, how did I not get, I made a nice slide that added a better label. So this is showing the DNA, then the RNA, primary transcript, and then the mature RNA. And I made a slide and I just didn't get it with that better label. So we start out with the DNA, and what this is showing is it's showing near the 3' end of the transcription unit. And it's showing that there's already some letters there that are going to be showing an enzyme where to cut the primary transcript and add poly A. And it differs in different genes. So we can know that in the DNA of the transcription unit for MYC, at that end, there's nucleotides that say, cut here and don't add very many poly A tail to this loser. So that's already in the DNA, that information to add the poly A tail there. So it's showing, identifying signal, like sequence to say, this is where you want to look, and then an actual cut site. So then this is the primary transcript, and you can see in DNA the T is a T, and in the primary transcript the RNA is a U instead of a T, and so it's RNA. It's got that same sequence, so it's got the site to recognize where to cut, and there's the cut site. And then here's the mature RNA, and it shows that it still has the site to recognize it, but it's cut there, and it's added poly A at the end. So that's a kind of processing that happens to the transcript. The transcript gets done, and it's initially just the same matching as the DNA, but it's got a recognition site, and the recognition site gets recognized, and the enzyme cuts it, and then adds a poly A tail as part of this RNA processing.
So I've got two questions. One
Oh, it's the 2-question move. Yeah, all right. My childhood pets were named. Oh no. Yeah?
Do you want to know the name of my childhood pet?
No, mine. Yeah, this
First
course is
question.
about me. If you haven't noticed yet, this course is about me, which none of us like. Yeah?
When we
it's
talk about
it's about
like,
my
a
pets.
chromosomal translocation that leads to an overexpression of MYC,
Yep.
Could that be because you said that it would, like, on the DNA, it shows how long the tail should be? Are you actually changing how
No.
long that should be?
No, when we have that MIC translocation.
Or just any translocation?
When we have that translocation, what we're doing is we're increasing the production.
Okay, not.
We're
adding
still
to the
making
tail.
an unstable MIC, but we're making more, so we shift the balance, right?
Is that
right.
a way we could in the lab make something more stable though, add on to its poly A tail?
What we could do is we could just swap this region, the MIG transcription unit, for any other gene, and it would have sequence here to say add a nice long poly A tail. Yep.
And my third question now, actually. So is it always the,
You have to pay more tuition in cash.
you guys pay me.
Oh yeah, that's right. All right. Well,
So is it always cytidine and adenine? Like, it's always CA?
yeah.
Okay, and we don't call it the CA tail, it's just the poly A tail?
Yeah, it's a poly A tail. Yep.
Okay, just no
Yeah. And let's see. Somebody else had a question.
Is this what they mean, how does this relate to the term like the 3' untranslated region?
Yep.
You see that like term UTR?
Yep, so let's look at that. So we glossed over that in this little structure. So notice here is a mature transcript of a coding gene RNA. It's leaving the nucleus, ready for the ribosome. It's messenger RNA. And we have the cap, the transcription unit, and the 3' poly A tail. And here's the coding region. And you can see that there's some of the RNA that's before the coding region, and there's some of the RNA that's after the coding region. The coding region is not as long as the transcription unit. And this is a good example to see what's going on. So in this part, this is called the 3' untranslated region. Great name. And in this region is where they have that binding site to recognize where to cut the RNA and add a tail. So the 3' untranslated region has significance. It has significance in the RNA processing. It has significance because we know in MIC, that's the region that's going to say, just add a short tail to this one, right? That's what, if we swapped with another gene, it would be there. Like that? Yep. Then here's the 5' region. It's called the 5' untranslated region. And this has interesting regulatory stuff. It's not the coding region. It's part of the RNA, it's part of the primary transcript, it's part of the mature transcript, and this has regulatory stuff too that we have not talked about. When the ribosome binds to this mRNA, the ribosome actually looks at this region, and there's some regulation of the ribosome that's present in that regulation. Remember, we said that MIC does not get translated well. The ribosome is like, I don't want to translate you. That's in this region here. So we could stabilize the RNA, make it last longer here. We could increase the translation efficiency of MIC by changing that part there, the 5' untranslated region. Yep. Yep.
And is that also where like the signal sequence is that marks proteins for secretion?
No,
and
the signal
insertion into
sequence
the-
is part of the coding region because the ribosome codes the signal sequence. It's a peptide sequence. So there are plenty of proteins that have to leave the cell or go to the cell surface, and a lot of those proteins, as part of the translation process, have an initial part of the coding region that codes for something called a signal sequence or a signal peptide that targets the ribosome to take it to the rough ER. Yep. But that's a introvio thing that we're not.
The ORF, I think the R and the F is reading frame, I just can't remember what the O.
Open,
Open reading frame, okay.
yeah, yeah, and we can talk about that, but yeah. Other questions people have? Okay, so great, right? So we see the structure of this mature mRNA, and we see, okay, 5' N, 5' cap, 3' N gets cut, and a poly A tail. And that's inside the nucleus, and the poly A tail plays a big role in stability. That's what we always think, poly A tail, and it's just going to get shortened, okay? The book, or those little summaries also show this, so we just want to look at this, where that image go. So this just shows an mRNA as it gets bound to the ribosome. And so one thing you can see is that there are these helper proteins that are going to help this protein, this mRNA, bind to the ribosome. And it said, the book, or the AI overview says, the 5' cap and the poly A tail, they're important for translation. And that's actually what the ribosome recognizes. And it's recognizing these proteins and how they're holding on to the 5' cap and the 3' poly A tail. No big deal, but just to show you that when the ribosome binds to an mRNA, it's binding to that. And there's a lot of potential for regulation there, to whether or not the ribosome is going to be like, okay, I see you, I'll translate you. Yep. So we don't have a specific example of that, but we know MIC is not likely to get translated. Well, it's because of issues like this, okay?
It's just, is it like that it's getting eaten before it finds it, or that it's like you're not very stable, we don't like you? Or is it kind of both?
I'll say both.
Okay.
Then, yeah, or you, Sarah?
I was going to ask, the ribosome does not bind to the 5' cap itself, it binds to the untranslated region?
Yeah, I think especially, right, is that these helper proteins are really what identifies the 5' cap and the poly A tail. And they hold it in this kind of folded manner. And I think the ribosome is actually recognizing the proteins more than the structure of the RNA. Yep. Yep. And I think this is the last concept of this, and that is that we talked about how the RNA gets degraded, or the poly A tail gets chewed up. The poly A tail gets chewed up when the RNA is getting translated. So it's just another detail that anytime the mRNA binds to a ribosome, it takes a bite out of the poly A tail, kind of like that. And a longer tail means you'll be around for more translation. Yeah, Mariah?
Can you say again which part of the ribosome binds to?
So I don't know, but almost certainly it binds to these helper proteins rather than the 5' cap or the poly A tail, because they're essential for it to interact with the ribosome. Yeah, and so there'll just be this little statement here I said like that. That just so you have a MIC example. So MIC exhibits a short poly A tail, is highly unstable. And then the second sentence says, this instability is linked to translation-dependent de-adenylation. So that's when the poly A tail gets shortened. Just kind of a little surprise, because you're like, okay, I accept that the poly A tail is getting, degraded, chewed up slowly. But it's really when it's bound to the ribosome. And so we'll remember that, because remember that MIC will bind to the ribosome, but then the ribosome's like, hey, I don't want to translate you. And as it's saying, I don't want to trans you, it's also chewing up the short tail. So it's kind of a double whammy, where you say, I'm going to do a short tail in the nucleus, you get out in the ribosome, in the cytoplasm, and when you bind to the ribosome, it's going to be like, I'm going to pause. And as it's paused, it's just chewing on that short tail. So kind of a double problem. In general, that's when poly A tails get shortened, is when the ribosome is holding it and they are getting slowly chewed up.
Is it the ribosome that has that exonuclease activity?
I don't know. Yep. Yep, okay. And so, see, poly A tail shortening is the translation dependent step in c-MYC mRNA degradation. That's generally true, that when the poly A tails get removed from any mRNA, it's when it's bound to the ribosome. Yep, and if you make it hard to translate, you're just going to shorten the lifespan of that RNA by holding on to it, okay? So let's look now at, that's what we need to know about those. Then another important thing that happens inside the nucleus is something that we're going to treat very lightly in our course. We're just going to move through this and explain it pretty quickly. And it's called RNA splicing. So that's our third example of something that happens post-transcriptional regulation and in the nucleus as part of RNA processing. And it looks like this, where if you look at the primary transcript, it's got the 5' cap, it's got the poly A tail, and then along the transcript, it has parts that are going to get removed. And those parts that are going to get removed are called introns, and their removal is called splicing. So we're going to splice out the introns, and we're going to save the pink parts, and the pink parts are called exons.
I
And
always
it-
hated that. Like that it should be the other way around. Like,
Shh. And that's just a fact of life. Like if you said, why are those introns there? No one has a good explanation for why they're there. Because there they are, right in the gene, and they are going to get removed. And that's just called RNA splicing. And we'll just show you that it looks like this, where here's an intron, and there are these protein complexes that bind to the ends of the intron, and they come together, and the intron gets removed. And when they come together, that's called a spliceosome. It's kind of a satisfying term. So it's very regulated, and it gets done. We're going to remove the introns. All right? And a lot of times that's just automatic. It's got to get done. We got this. And the introns are considered just junk, and they're going to get removed, and they just get destroyed. But having these introns provides a potential for regulation. The regulation is that we can choose to eliminate introns or not, as it's regulated. And we see in more complex organisms, so meaning like comparing a human to a fruit fly, we see more of this regulation of splicing. If we looked at the fruit fly genome, we would find that the fruit fly has about 14,000, 15,000 genes. And the fruit fly genome was sequenced, and that was known as a fact about 20 years ago. And the human genome was not sequenced yet. And at the time, people were like, we'll probably have about 100,000 genes in humans, right? Because if it takes 14,000, 15,000 to make a fruit fly, they have a lot of genes, right? So the human genome was sequenced a few years later, and we only have about 20,000 genes. So you go, well, how are you going to explain that? How can you explain that, like, I think a yeast that causes bread to rise, that has 8,000 genes. So we have like 3 times more genes than yeast to make a freaking human, right? How's that work on our planet? And some people love to talk about this because there's a lot more of this regulation of splicing in humans than there is in yeast. There's kind of none in yeast. And there's a fair amount in fruit flies, and lots in us. So we've talked about genes like the estrogen receptor. The estrogen receptor has lots of these introns, and they get removed in a regulated way. And here's an image of this to show you this. So let's just look at this because it's nice, I think a nice color-coded image. And this is a place where I've also added the labels. So this is the transcription, that's a unit, that's the DNA. Here's the primary transcript, and here's the mature RNA. So we'll focus on here, the splicing. And it's going to remove these yellow parts. Those are the introns, okay? So what you can see is the transcript that we've always been drawing. That actually includes the coding region, right? It also includes, it can include these introns. And there are specific numbers and locations of the introns. And they're going to get transcribed, and they're part of the primary transcript, but they're not part of the mature transcript. So that's this RNA splicing is in the nucleus. RNA processing includes the spliceosome removing the introns. And the mature RNA is made up of just exons, okay? And so you can see that then leads to the protein. The protein is got coded for by exons, but not the introns, because they got removed in the nucleus, right? So that just shows you this general idea of splicing, and then the protein is made up of these individual exon units that got joined together. Okay? So then, oh man, here is this regulation, and it's called alternative splicing. And so I just added this figure to make it look like this. So we just looked at the example where 4 exons got spliced together to make this protein. For the estrogen receptor and probably every gene we talked about in our course, there's this alternative splicing that's regulated. And at some point, the cell doesn't add all the exons. And it's regulated, it's not random. So we saw, we're supposed to add 1, 2, 3, 4. But sometimes the second exon gets spliced out like it's an intron. Or other times you can have this, where you get the first and second, but maybe the third or the fourth. And so you can see we're mixing and matching these, and we get proteins out that are different. And the way to think about them is, here's a protein, it's like your body, and you've got a head, you've got a trunk, and you've got legs and arms. So just imagine it's you, but no head. Or you and no body. Or you like that. So alternative splicing, where the product that is the protein is exactly the same in some parts, but then it's either missing or has different other parts. And the estrogen receptor is like that a lot. And when we have that, those alternative products of the same gene are called protein isoforms. Yep.
Is it the spliceosomes that also remove the exons for
Yep.
this isoform?
Yep, so we have that spliceosome activity that's regulated to affect this alternative splicing. And we don't want to go deep into this in our course because it's just an example. But a lot of people think this is what makes humans fancy compared to a fruit fly or a yeast, is lots of this alternative splicing. You don't need more genes, you can splice them differently. Now, are these paralogs? No. We learned about paralogs, and paralogs are multiple genes that are slightly different. This is one gene that is exactly the same. It's me, right? Paralogs would be a group of people, right? Different people, different genes that are really similar. This is one gene, and we make these different products that are exactly the same except for little chunks here and there, and they can create a more complex functional protein. Yep?
I just missed a word that you said. You said this is what
I
makes...
said party.
This is what makes humans what?
Party.
Oh, okay.
Nice. Yes. Fancy.
Okay.
Yeah, get that in your notes. Fancy and party, right? But fancy compared to a fruit fly, We don't have that many more genes. How do we get there? And the answer is maybe they just took the individual genes and made them more complicated, like that. Because there's a lot more of this in us. And we are just not going to go down to the details of what do the different estrogen receptor isoforms do, because they must do very similar things, but slightly different. One of the things that estrogen receptor does is it binds to the DNA, right? It's got a DNA binding domain, it's got a zinc atom in a zinc finger DNA binding domain. Some of the estrogen receptor isoforms don't even have that, or they have a slightly portioned out different one, So they might bind to some different target genes, or they might not bind to any gene, and they might be some sort of inhibitor, and blah, 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 So it gets into the details of each protein's function of what these different isoforms do. Key concept for us for the final exam is alternative splicing of one gene leads to a variety of proteins that are called isoforms. Evolutionary gene duplication leads to multiple genes that are called paralogs. So it's a different story, right? One gene, alternative splicing, protein isoform, versus multiple genes and a gene family, paralogs. Both of them are, the result of evolution through natural selection, but they really target a different part of the DNA that we inherit. Yep. Okay? So that's really all we want to get out of RNA splicing. Just that example, and really boiling it down to appreciating the idea of alternative splicing. And it means that we can regulate that intron removal. And sometimes we can include an exon and remove that. Or other times we're choosing between two exons that are side by side. In a certain case, we'll pick this one. Okay, so we'll pick that one. So it's me, and in some cases I don't have a head, sometimes I have my head, sometimes I have somebody else's head. And it's still me, but it's like, whoa, Dave, or Jennifer. Yep, okay, so that's RNA splicing, and good one, right? So famous one. And then the last one is something we have talked about, and that is RNA interference. And RNA interference always reduces the gene expression. So it's a way of knocking down the gene expression, and that's the term people use. When we mutate the DNA, we can destroy the gene. That's called a knockout, because we have permanently destroyed the gene. We've knocked them out, like a fighter that knocks the other boxer out, and they're not going to get up. When we inhibit gene expression by RNA interference, what we're doing is we're blocking gene expression, but not by affecting the DNA. And so that's temporary. If we stop interfering, the gene will be normal and everything will be fine. And there are really two stories of RNA interference. One happens inside the nucleus as part of RNA processing. The other happens in the cytoplasm as part of translation. So there's a good chance you've heard about RNAi before, and it's always used to decrease gene expression. And there are two stories to it. And the first story uses siRNAs, and it causes the RNA to get destroyed before it leaves the nucleus. So it's part of RNA processing, and it causes the RNA to get degraded before it even leaves the nucleus. So that knocks down the gene expression. And so we just are drawing a separation here, right? So we're talking about a certain type of RNA interference. It's siRNA interference that happens inside the nucleus as part of RNA processing. Before it leaves the nucleus, it could get destroyed. The next thing to say about it is that this is unusual in nature. So out in the real world, it happens, but it's unusual. Scientists, when they discovered it, they realized they could take advantage of it, and they could design transgenes and use this type of RNA interference to destroy any gene product and reduce it. So this is a very famous example because of how it's used in research labs to destroy the RNA. And scientists like to do it that way because it's easy for them in an experiment to measure. Before they did their experiment, there's the RNA. After, the RNA is gone. So it's a really easy experiment to do to confirm, yep, our RNA interference is working. So again, in nature, but rare in nature, and used in research labs a lot, right? And so there'll be just a little thing here, and we'll just talk about this. So it's siRNA, and it has a feature where it's going to make a perfect match. And what's going to make a perfect match is you have a target RNA, like a messenger RNA, in the nucleus, and there's a short complementary RNA they can base pair with it. The short RNA is called an siRNA, for small interfering RNA. And it binds, and that double-stranded RNA gets targeted for destruction. So it just says it interferes with the expression of specific genes with complementary nucleotide sequences by degrading messenger RNA after transcription, preventing translation. And this image we don't need to know about, right? We're not looking at this from our book, we're just learning about it in class, so keep it pretty straightforward. siRNA works in the nucleus to target an RNA for destruction. And an siRNA is just a short RNA, it's only about 20 nucleotides long, to make matches. Yep?
Are these the same thing as microRNAs?
No. MicroRNAs are going to be the second type of RNA interference. So we have two types of RNA interference, and since you asked about that, Cam, we'll just look at this, because it's really important for us to distinguish them, right? Because remember, our classroom is the nucleus, that's a nuclear pore. siRNAs are in our classroom, and they're going to bind to the target and cause the target to get destroyed. And we can design an siRNA in a lab to target any gene. So let's say we're studying the insulin gene. We could design an siRNA transgene and cause it to destroy the insulin RNA before it leaves the nucleus. So that's siRNAs, they're in our classroom. We're going to talk in a minute about outside our classroom, in the hall, in the cytoplasm. And that's where microRNAs work. And they work by binding to a target and blocking translation. So just to kind of move in that direction, let's say we're studying insulin. And we're like, we want to study insulin, but we don't have a mutation. We don't have a knocked out gene, so we don't know what it's like to have a mutant with no insulin. So we're going to design in lab in a test tube an siRNA that will be a perfect match to the insulin RNA, and we'll express that. And when the cell expresses insulin, it'll get transcribed. There'll be RNA processing occur, and our little siRNA will be in the nucleus. It will base pair perfectly with the insulin, and it will target that for destruction. That we choose that because it's good for us to study it that way, because we're like, before we did our experiment, we could detect the insulin RNA, but after our experiment, we couldn't even detect it. The other is we could study it this way, where we could design a microRNA. And a microRNA has a different sequence to it. It binds to the insulin RNA, but doesn't bind perfectly. And the microRNA is actually out of the nucleus. It's out in the hallway. And we'll talk about it in a minute, but it's really important for us to distinguish between RNA interference that takes place in the nucleus and by siRNA and leads to destruction of the RNA versus, and this will be fun because people will be like, well, where'd Dave go? Versus microRNAs that are in the cytoplasm, bind to the target insulin and block it from translation. The effect is the same on the cell, no insulin protein. Are we missing insulin protein because it doesn't even have the insulin RNA, or because the insulin RNA is there, but microRNAs prevent it from being translated? Yep. And most of the time in college, people just talk about RNA interference. And they just say, they usually just talk about it getting degraded. Because that's, the science one where they're doing research in lab. In nature, this one, microRNA is super common in all kinds of plants and animals. These are more common in more complex organisms, so it's another example of maybe this is how we got fancy, But again, we're distinguishing that here because we're inside the nucleus with the small interfering RNAs and destruction of the RNA, or we're in the cytoplasm with the microRNAs and blocking translation.
Dave,
Yep.
you just mentioned that siRNA binds perfectly, but microRNA binds imperfectly.
Yeah.
Can you elaborate a little bit more on the importance of that distinction?
No, because I don't want to, and also because I don't really get it either. I mean, I get it, but that's what they're showing down here is they're getting into the nuts and bolts of how the siRNA gets prepared for use and how it differs from a microRNA. And so we can think of it this way, right? These siRNAs and the microRNAs, they're examples of non-coding genes. There's a gene, we make a gene, or there exists a gene that codes for this. And they always have a transcription unit, and the transcription is longer than the mature process. So I'll just scroll back here to show you that. Down here it says, we're especially focused on the coding genes. Down here it says non-coding genes also have RNA processing. So not to confuse people, but the story of siRNA and microRNA also includes their expression and their processing. And their processing includes a kind of a complicated story that differs between siRNA and microRNA. And for our course, we don't care. And partly because I don't want to overburden it, but also because I could explain it some, but for years and years, scientists were pretty frustrated and confused about the relationship between microRNAs and siRNAs. And I think that because of their naming and stuff, this is still a little bit of a loose categorization, but it's becoming more distinct because people are like, let's make sure we call this what it is. So it's either an siRNA and it's got perfect base pairing, it's processed as an RNA that way, and it's going to cause degradation. versus microRNA and how it gets processed and it causes block in translation. Our key thing is that physicality of it is siRNA's function in the nucleus to degrade targets and microRNA's function in cytoplasm to block translation. And so just a little bit of small interfering RNAs, and just to read about it, small interfering RNA is also similar to the miRNA, which we'll talk about again a little bit from now. However, microRNAs are derived from shorter stem loop RNA products. MicroRNAs typically silence genes by repression or translation, have a broader specificity of action, while siRNAs typically work with higher specificity, including RNA before translation. So they're talking about that there, and we're just going to talk about it. Look, it's pretty well ironed out, and we got siRNAs and microRNAs, and it's getting pretty well ironed out. It's pretty clear that there are two types of RNA interference. Yep, and that would be great, because again, we're in the nucleus, and we're talking about RNA processing, and so we're still at the level of inside the nucleus, post-transcriptional regulation, this RNA processing, and we have these four favorites: the capping, the poly A tail, the splicing, and the RNA interference by siRNAs and degradation. Cool? So see, we moved it down here. We say after the mature RNA leaves the nucleus, RNA interference occurs by a different mechanism using microRNAs, miRNAs, that block translation. And so we'll have a concept chat after we just look at this one thing. Just read a little bit about microRNAs, not to be confused with mitochondrial RNA. So, miRNA. MicroRNAs are small single blah, 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 silence said mRNA molecules by one or more of the following processes. So see this where it gets a little crappy for learning, because they can cause some cut, but not like a degradation cut. They especially do it by reducing translation. So again, it doesn't fall out with perfection, but it's falling out more and more in a more satisfying way for learners, but also for the reality of nature, And it's been confused and kind of conflated for a long time. Yeah, and it was super confusing at the beginning because the the small RNAs were like, this one's different, and it seemed to get processed in a different way. So it was confusing for a long time, and it was like we're talking 25 years ago that it was confusing, and it's kind of slowly getting shaken out. So we're in good shape learning it the way we are learning it here. There's a question here and that and there.
I have a question about the poly A tail. We're in the cytoplasm now, right? So why are we the poly A tail still needed in the cytoplasm?
Yep.
I thought that was just to stabilize it when it was in the nucleus.
No, the poly A tail is to stabilize it for the life of the molecule, either in the nucleus, but also in the translation. And remember, it's translated is when it starts to get shortened.
Okay.
So it's pretty stable in the nucleus,
The poly A tail, okay.
except like the poly A tail is. If it doesn't have a poly A tail, it could get degraded pretty fast, but it has a poly A tail added. Yep.
So you really just want us to know that microRNA just binds and blocks from translation?
Yeah. Let's take the final exam. Put your books away, and we'll have like a 5-question final exam, and we'll get out here and go swimming. Yep, exactly, right? It's a great thing to do to just repeat your understanding of the thing, and you could say it to me, or you could say it to a pet, right? And say, this is what we learned in class today, Ginger. Like that, right? But yes, absolutely, right? There's a take-home message here, and it's a framework, right? There's a lot of different examples of RNA processing. Let's build a framework so we have a conceptual appreciation of that and some examples of how it gets regulated. Yep. MYC gets targeted by a microRNA, LET7. That was in our textbook. It was on our exam. It was the one question on our exam that was a hard question because it was just from our book. But our book described it and had a little figure. So see, poor MYC, right? I call it a loser because it's just getting pounded on at every angle, right? And remember that there was this question earlier today, right? When we have a translocation, we still have all of those issues with MYC, unstable, not translated, has a microRNA targeting. But what we do with the translocation is we just increase the production to a sick amount. And the degradation stays the same rate, right? And you adjust, the equilibrium. You're gonna have more MYC if you keep the degradation at the same rate and you just crank up the production. You're gonna overdo it with the MYC. And we don't wanna do that because it just lights up the cell cycle. Yep. Okay, so see, satisfying, right? And Yeah, we're not trying to confuse people or, you know, start causing you to unlearn. Just kind of fit it together because that's an important concept for that. Yeah, we learned about translocation, right? We didn't learn about any mutations in the MYC transcription unit. We could see that that could happen, right? It could affect the poly A tail. There could be a mutation there, maybe make a longer poly A tail. We haven't seen that. Maybe there are examples of cancer, but the big famous examples of cancer where you just crank up normal MYC and you just overwhelm those downstream unstable elements of the MYC story because you're just making tons of it and you're degrading it fast, but not fast enough. Yep. All right, so let's do a concept chat and see what you thought was important today. And, you know, talk to somebody else. Good time to say.
Learned about like the four main
What's your favorite one?
probably the RNA splicing knows that that was what I want to write about, but I guess I have the most background knowledge on that.
I think we did a drawing section or like what part of it?
It's always bothered me. It's like cut out, like they use the answers, like that's always been bothering me about whatever. So yeah, so like you keep the end, like
The exons leave
yeah, they leave the nucleus, like so like well I guess that makes sense too, but like you cut out but they're just like so also the poly tail, which is a whole bunch of adenines. One thing though, so like, and I think just cleared up is that the shortening event occurs not in the nucleus, that occurs in the cytoplasm.
Right, because when the complex binds that has the ribosome model, that's it. And that's what he was saying today, is that it tries to bind, but it's not stable. And it's trying to bind, and it's still very true. It's like if the tail's in the timer or something.
So this is kind of like when the where he was binding to the 27 and it was like a time bomb. But I also was really happy to be that because like I remember being like, what do you mean that the ribosome doesn't want to translate? Like these things don't have brains, how can they just be on like petty but like that's good to know.
Something that I kind of wondered is
I was confused about
when the tail is aligned, does it start eating like slowly like so could you get like 1, 2, 3 but not 4? Would it eat it up so it goes forward and it
I think that once it starts eating that, I think that is like
that might be a 2
yeah, I mean, like, I think it's like, pushing, like, maybe we would start eating one, and so, like, would it still make an alternative gene, like that isoform? But I kind of feel like you're then eating the coding region, and you're going to lead more so to not one full gene being cut out, but, like, a mutation. You're only going to get it. So I wonder, it sounds to me like, it seems to me that if you were in there, you should just be done.
I think the open is over.
The open reading frames, like, you're after
That's the code region.
yes.
Oh no, so like alternate splicing is different than parallel because alternate splicing is the same gene, the same person. Yes, parallel splicing is the same gene, and then the second one changes. But it's the same gene, just reorganized. Same gene, different.
And then we learned that for RNA interference by the siRNA, that's the nucleus. And that is 100% and it's going to decrease its normal expression. And then the miRNAs are in the cytoplasm, and that is going to block its translation. And I think that's all we've actually learned. And then the LET7 is the microRNA. This one's common in nature, right? Yeah. Yes.
I think that
We'll just do this for another minute.
siRNA is something
And then we will check in with Lilith. What's one thing your group talked about back there, Lilith?
We talked about how siRNAs they destroy their targets in the nucleus, and then the mRNAs block translation of the cytoplasm.
Yep, awesome. And let's see, and the next person I had down was Teresa. Let's see, oh, maybe Teresa's not here. Okay, so how about Rustam?
We talked about the poly A tail, and the longer the tail is, the more stable it is.
Oh, yep, yeah, and and that's I think a general rule, and I bet there are some exceptions to that, but it's a good, general rule. And in general, they're like plenty long. They're like a couple hundred nucleotides long. And when it's regulated, it's like a mixed story where it's like, oh, you're going to start with a short one, and you're just not going to be lasting very long. Yeah, Lydia.
We were wondering for the siRNAs versus the mRNAs, can it like unblock transcription in the cytoplasm, or is it like block, shut down, and it won't ever?
It's there's this official term that people use, and they'll say when a gene has a mutation, it can cause the gene to be knocked out. It will never function again, not in this organism and not in any of their progeny. So that's a knockout. And they talk about RNA interference, whether it's siRNA or microRNA, as a knockdown. It's temporary. If you remove the microRNA or the siRNA, the gene expression will return. Yep. Yeah, okay, great. And then, let's see. So Lima, what's something your group talked about too?
We talked about also alternative splicing, but how that's different from gene duplication. So alternative splicing is when one gene needs two isoforms, and gene duplication is when it's paralogs.
Yeah, and that's just an important distinction for us. That's something that, when we talk about alternative splicing, that's one of the most important things for us is to understand that it's talking about a single gene and evolution of that gene to make more of a complex set of products that are called protein isoforms. Yep, and we just want to be sure we understand that distinction. Great. Do people have any questions? Yeah, Sarah.
We talked about translation and how, basically wondering if it's one ribosome, like at a time that translates the mRNA, or can it be multiple at the same time? And then,
Yep. Yeah, so something we haven't really talked about very much in our course is we're always talking about eukaryotes. And in a eukaryotic cell, it's one ribosome at a time. And it's in the cytoplasm. So, in eukaryotes, we always have a nucleus, and we're separating these steps in gene expression. This is not a class in microbiology. And a lot of people get confused about that because, molecular biology, microbiology, they must be closely related. Molecular biologists deal with a lot of microbiology, but microbiology is really talking about single-celled organisms and especially prokaryotes. In a prokaryote, there's no nucleus, and what you see in prokaryotes is as the RNA polymerase is producing the transcript, a ribosome binds the transcript as it's being made. And as it's being made, another ribosome will bind, and another. So it looks very different in a prokaryote than what we're learning about in our course in a eukaryote. And it looks different, but it's the same kind of story. We're going to transcribe the gene, and we're going to have the ribosome translated. It's just that we don't have that compartmentalization, and we don't have that amount of potential for RNA processing in prokaryotes. Yep. Other questions people have? So, I put in a couple slides like this. We don't really need to look at it because when I was explaining the siRNA and microRNA, I could see a couple of students were kind of frowning because I assume, you've learned about these in other courses, and this seems like a kind of maybe something new or a surprising distinction. Here's a great way to understand this distinction forever and experience me crying at the same time. So back in about 2001, these two scientists who were not famous scientists at all won a Nobel Prize for RNA interference. And the world was shocked because they won it for medicine, and the two scientists were in different labs. One was in Worcester, hello, winning a Nobel Prize, and the other one was at Harvard. But they were studying this nematode, and it does nothing to do with medicine. But they really won the Nobel Prize because it was a natural process, but scientists realized they could take advantage of it, and they could design these small interfering RNAs and knock down any gene and use it in a research lab to quickly make discoveries without looking for mutations, right? So that's like, over 20 years ago, these two scientists won it. And at that time, there were other scientists that did not win the Nobel Prize in medicine, and people were shocked. There were other nematode biologists who did at least as much work as the two people who won it. And it was super confusing like that. And again, they won it because it was a natural process, and they were really studying that siRNA and that destruction of the RNA, right? Like that. So they really won it for those two purposes. Yeah, it's natural, it occurs in nature, occurs in humans, but especially, we could take advantage of this thing and really exploit it, right? Like that, okay? So one of the scientists that didn't win a Nobel Prize was named Victor, and my friend, one of my best friends, Ann, was in Victor's lab. And those guys were so ticked off, like that, right? And then there was another guy, Gary, who also studied the same thing, nematode, different topic in nematodes, but who cares about nematodes? They were just discovering a natural mechanism. Gary didn't win a Nobel Prize either. And all the people in Gary's lab were so ticked off and just freaked out and mad and made lifelong enemies by slashing tires and all kinds of crazy stuff. Yeah, my friend Ann totally crazed out by it, because her boss didn't win a Nobel Prize and they were doing Nobel Prize work, right? So last year, Gary and Victor won the Nobel Prize. And they won it for microRNA, because that's what they were working on. And apparently, and I don't believe it, but apparently the people who picked Nobel Prize had that kind of insight, where they're like, we're really focused on it's natural, and you can use it as a tool. So we're going to give those guys the Nobel Prize in 2001 or about then, And they just didn't even mention that, we might give a Nobel Prize another time, maybe 20 years from now, you guys could just settle down, But it's just a startling fact, like in science history, that there was that, these two awards. And it's a good way for you to appreciate, What was really exciting initially is, it's a natural thing, but we can exploit it in lab, and it's just exploited. In that nematode, people quickly made siRNA genes that could knock down any gene in that species. And they made them and they just froze them, and you can just order them. If I'm studying gene X in nematode, I can just order an siRNA construct that will knock it down. It was just a hugely significant and exciting tool, right? So they won a Nobel Prize. And these other people were just shocked. Here's another group of people that were shocked back in 2001 or wherever it was exactly. There were these people who were studying petunia flowers, and they had discovered also microRNAs, and they were really mad because they were way ahead of the nematode people. And at that time, people thought, well, you're not going to give a Nobel Prize in medicine for somebody who studies a flower, because the general public would just be like, what? Science is stupid, right? We can't give them, they're studying a flower and they're like, yeah, you saved some lives, right? And so that's what they thought was going on, But the thing is just that they were studying really the microRNA, right? And so now you see it kind of playing out in history now. People, even the scientists, people are appreciating more and more kind of the distinction. And again, it's not perfectly ironed out. It was just last year that the Nobel Prize showed up for those microRNA people, like that. But it was so weird when they won that first Nobel Prize. Like my friend Ann, she was just crazy. And she's still bitter, I think even after Victor won, I think she's still bitter about it. She's just like, bah. But anyway, we have to stop. So again, we don't need to focus on that. We're solid on our processing and that, and we're going to move on Tuesday to some more topics. Yep. Awesome.